strategy here will be to talk with you about the new genetics. I will raise questions primarily. I believe it was C.S. Lewis who said the best meeting he ever went to was all questions. I want to share with you some theoretical scenarios which come from Science News, an article entitled Predisposition and Prejudice. The first tells about Ellen, who spent 4 years completing her PhD in engineering. Now applying for a job, the company doctor drew some blood. She could hardly contain her excitement about the job she'd been offered at one of the country's foremost metallurgical research institutes. 2 days later, a phone call came. "You are perfectly healthy," the doctor said, "but tests have revealed that you harbor a gene that can result in decreased levels of a blood enzyme, glucose 6-phosphate dehydrogenase. Without the protection of that enzyme, you have a slightly increased risk of developing a red cell disease if you come into contact with certain chemicals in our laboratory." "I'm sorry," he said. "The job has been offered to someone else." The second scenario. When Frank got married at age 31, he decided to take out a life insurance policy. He was a swimmer and a racquetball player and had no previous hospitalizations. He felt certain that his low premiums would be a good start on a family. Weeks later, after a routine physical, the insurance company responded. Sophisticated DNA testing had revealed in his tissues a single missing copy of an anti-oncogene. An oncogene is something that causes cancer, and this is something that prevents that. And there were some other minor variations. Computer analysis showed that these misprints in his DNA more than tripled his risk of getting small cell lung cancer by age 55. His application was rejected. Now, those are not real events, but they're not implausible. Discrimination of this type has occurred before, but now it can become more extensive and more precise. Knowledge about ourselves and our susceptibilities can hurt. But who else should have access to this information? We have laws protecting the handicapped. But is a person currently perfectly healthy, but with a susceptibility, a handicapped person that will be governed or controlled by these laws? Employers indeed must be concerned about healthcare costs. They would ask you to sign a waiver to your medical records. Do they always have the right to this information about you? Some have said that this new concept, this extension of diagnosis, may force a total rethinking of the concept of health insurance. For insurance is against the risk of the unknown. The more that becomes known, the less there is to insure against. It is also thought possible that individuals who become aware of their own personal risk may then immediately go out and ask for health insurance. Well, this is the whole cluster of questions that come up. In a broader sense, genetic engineering and biotechnology more generally are changing what the very word life means. The essence of life in this new context is seen to be its constructability, the ability to change. Furthermore, biotechnology has a strong appeal as a technological fix and as a means of salvation for our economy. As a result, biotechnology has become big business. Thus, the message is clear. Biotechnology is in the process of changing our views of life and perhaps also many other features of our culture. I'm going to be showing a series of slides. Rather than turn the projector off at all times, I will leave sometimes a slide showing while I want to talk with you now about what DNA is like and some of the information concerning that. I'll go on to something about gene mapping and further implications. What do we know about DNA? How much is there, for example? In one set of human chromosomes, there are 3 billion nucleotides. Each one of those is a bit of the genetic information. It's of some interest that this approximates the number of individuals in the population. And so at the bottom you see numbers of people, 3 billion, about, to give some idea of scale. These then we then find about 50,000 genes. Perhaps there are 50,000 towns and villages in the world. Genes are on chromosomes. There are 23 pairs of chromosomes. It is said that it would take about 13 volumes of Encyclopedia Britannica if it were printed without any punctuation to include the amount of information in one set of human genes. This is an enlarged human chromosome. If a drug had not been applied, it would have split down the middle and become 2 new chromosomes. You see some little things sticking out of the edge, and we will enlarge them. In the center now is that scaffolding. There is a halo of DNA that has been relaxed and permitted to flow out into the medium. Let's take an enlarged view of this. At the bottom is the scaffolding, and then there are loops of DNA. A chromosome then is a large single long molecule of DNA which is coiled and coiled and coiled. It is marvelous and wonderful that it's possible for this to replicate and then separate without getting hopelessly tangled. So then you see strands of DNA. It's impossible to say what gene is there. It all looks alike. One section of that conveys the information for a particular protein product is a gene. And all of this is organized around chromosomes. Now the basic story that started all of this goes back to the year 1953. This was the year that Watson and Crick suggested a model, a way of thinking about the structure of DNA. Prior to this time, it was known that the molecule is a double helix. It is something like a very small spiral staircase. If you go up that staircase and look at the stair treads, you notice sort of toward the middle, on 2 sides of the middle, there are some letters. These represent the bases. There is a particular pattern of pairing which is essential. As a matter of fact, Watson and Crick said, we think that's the way it has to be in order to account for the fact that a gene must be duplicated correctly and it must convey information. You'll notice that in every instance there is an A opposite a T. That's a double bond, and that's why only those 2 will fit. G is always opposite C because that's a triple bond. Thus, when the 2 sides split apart and other bases slip into place, you get 2 precise duplicates. As you walk up and read the code along one series, from step to step to step, there is a code which then specifies the sequence of amino acids. Now, a little over 12 years ago, the further steps for recombinant DNA technology began to be used. This resulted from the discovery in bacteria of enzymes which have the property of cutting up DNA. This is a way that bacteria can protect themselves from invading viruses. Each of these enzymes will detect a particular genetic code cut at that place only. So to the left, there is an illustration of a ring DNA. An enzyme has cut it at 3 points. These cut pieces of DNA are put on a gel-like medium, subjected to an electrical charge. The smaller the piece, the farther it will go. And thus you see the C segment, which is the shortest, has moved the farthest. If a different enzyme were used, it might cut at 5 places. And again, they would move depending upon their distance. So we have a system now for cutting up the DNA in a repeatable kind of manner and sorting out pieces. We now have to have a way to recognize one piece from another. If I were to do a DNA study for you, blood sample would be drawn. The red cells are of no use because they have no nuclei. But the white cells do have nuclei and thus DNA. The DNA would be extracted, it would be cut up with enzymes, these would be put on the gel, the electrical charge applied. Then over this would be put what is called a probe, some radioactive mirror copy of the DNA. This would be so selected that it would recognize only the gene that makes insulin, or the gene for hemoglobin, or some other particular kind of gene. Now we've learned some things about the DNA from this kind of process. As a matter of fact, one of the important things about recombinant DNA technology is that it has drastically changed the science of biology. One of the features is that gene splicing is not something that the geneticists thought of. It's been happening in cells as long as we can imagine they've been around. Up at the top is a section of a gene. There are blocks that are labeled as exons. These eventually will give rise to the pattern for the gene product. There are other pieces called introns, which are edited out and removed. If there is an error so they're not cut out, the gene will be imperfect and not be able to function. So you see the next to the bottom is a messenger RNA. There are still left 2 tails. In the tail to the left, there is further information which says at what time, in what cell, and in what amount this particular gene will be expressed. The other tail has another function. Then finally, it's translated to give a protein product. It's given us some further information about developmental process. This is a chromosome which has genes for 1/2 of the hemoglobin molecule. There are some black bars there. These are read one at a time. In development, first the far left one, then the next, then the next, and then the next. So there is a time switch mechanism. As a matter of fact, some individuals have a mutation of the switch. Therefore, throughout their lives, they will make fetal hemoglobin rather than adult hemoglobin. The point here is that no cell reads all of the genes at the same time. There is a part of the program which is reserved for this. In your computer, you can imagine that there is a program which never shows on the screen, but it controls what does show on the screen and when. How has this information been growing? Victor McCusick has an atlas of genetic traits. The first atlas had very, very few. The big, thick book that we have now has well over 4,000 identified genetic traits. We also can now develop a genetic map. This is so complex that the slide can't possibly show it. I have, and I will bring tomorrow, a copy of this map. This is now dated, so I have a map as of February 1989. And if I want to have more, I can dial up on a computer to New Haven and find the latest word. To the left there, you will see at the top chromosome number 1. These are numbered by length. At the very top there, you see RH, the RH blood type. on chromosome 6 to the bottom, there is a gene. I have been involved in mapping one of the kinds of cerebellar degenerations. Now let me tell you just briefly about a paper that we have published, which I have not yet seen in its final form. It's due out this week, and if you took the journal Nature in your library, I suspect you haven't, the copy would be there for the first time. We have mapped a gene for this particular condition, benign familial neonatal convulsions. It follows what's called an autosomal dominant pattern of inheritance. For every affected person, the risk to a child is about 50%. Large pedigrees can be obtained. Onset is usually in the first week of life. At about 3 to 5 days of age, the child will begin to have convulsions. There may be 15 to 20 a day, each one of them lasting about 2 minutes. Severe enough that there will be some lack of breathing and some bluish color begins to appear. You can imagine how frightening that would be. Not to worry, it will go away. And the family needs to have that information so that they will not be overly worried. This is the pedigree that made the difference. The white symbols indicate persons with the trait. Now there is dimly shown there a kind of purplish color. You see that below some of the symbols? That represents a marked chromosome. The group in Salt Lake City who worked with these blood samples were prepared to use 250 different genetic probes stretched out over the human chromosomes. When they got to 92, the job had been done. One of these is called a VNTR, a variable number of tandem repeats. There is a short segment of DNA, which in some individuals is only one piece, sometimes it's 2 or 3, or up to 16 segments of this. And as you can guess, the more there are, the longer it is, and the different position for the band. It turns out that every person affected in this family has an 8 type for that marker and a 2 for another marker. So this then is not, these are not the genes for the disease, but they faithfully track the disease through the family. Is that point acceptable there? So now we have a marker. There are 3 individuals shown with asterisks. These individuals must have gotten the marked chromosome, but did not develop the convulsions. And this may represent what we call lack of penetrance. The gene was there, but was not expressed. But now it's clear that both of these markers are on chromosome 20. When we put this all together, the odds that this gene for the disease trait is on chromosome 20, 450,000 to 1. And that's pretty good odds. Here is chromosome 20, and that somewhere in that area is the place where this gene is located. Now, we know that other genes have been mapped, and this process is proceeding very rapidly. I'll talk more about Huntington disease at the moment. Cystic fibrosis is another one on chromosome 12, phenylketonuria or PKU on, CF on 7 and PKU on 12. One of the problems is, if you know it's there, it may be very difficult to actually find the gene. Laboratories are convinced that they have in some test tube the gene for CF, but they don't know what the code is. And you could walk right along the chromosome and go right past it. It's like going through a whole series of letters without punctuation saying, when am I there? It's not as easy as it may seem. We know that in some families, a manic depressive disease called bipolar affective is on chromosome 11. In others, it's on the X chromosome, and in some others, it's on neither one of these. Now, how can this information be used being of help to people? It's of little help in the disease I'm talking about. No one would ever do prenatal diagnosis for such a benign kind of condition. but I will talk with you about Huntington's disease. This is the thing that Woody Guthrie had and died from. It is a serious disorder. Once it sets in between age 10 and age 70, within perhaps 15 years, death is certain. You can see at the upper left a filled-in symbol, and there is a sort of fine print above that, a marker for a gene defect, and a marker locus A. The other chromosome in that person is marked B. At the bottom, the lower, the one to the left, has the defect and an A chromosome. The next one is normal, has a B chromosome. But in the middle is a person who has a defect with the B marker. This represents a crossover. Now that we know that the marker for Huntington's disease is on chromosome 4, if you had a parent with this condition, you would be at 50% risk for developing the disease. Would you want to have the marker test done to find out not a 50%, but 95% versus 5%? Would you like that information? Here was a group of 55 people who are actually at 50% risk. Of them, 40 said that they would take the test. The others would not. What if that test were positive? Would you have children? Each child then would be at 50% risk. 13 of those said that they would go ahead and have children. What if prenatal testing were available? Would you have that test? Again, some would and some would not. What if the prenatal test showed that the child carried that gene and would be sure to develop the disease? Some would have termination and some would not. We cannot always predict what people will do. If you as pastor have someone in your church or someone else in the community who is in a position for having an option for this test or actually having it, would you be able to talk with them sympathetically and understandingly about what this does to their own self-image? This now talks about a different kind of problem. This actually has to do with hemophilia. Hemophilia is a very tragic disease. And it was asking about whether people would be willing to have another child with such a condition. And you'll notice here that those who have been exposed to that by having a prior child, 9 out of 11 would not want to take the risk. Others who find out, other women who find that they're a carrier but have had little exposure to the disease, are much more willing to take the risk. Which is worse, to have a child with a serious disorder or to have no child at all? What about using selective abortion? Here you see that for those who said the greatest loss would be to have a child that's affected, they would tend to favor selective abortion. But those who were more worried about having no child, their opinion would go in the opposite direction. Now a few comments about the implications of this new information about human sexuality. Sexuality is a gradual process that begins in the fetus and continues in postnatal life. The sex of a fetus cannot be distinguished before 6 to 7 weeks of life. So there is an early bipotential gonad that moves one way or another. There is a factor called a testis determining factor that makes the difference. If that is absent, then the female will develop. The default developmental pattern in the computer sense is female. It is now known that this testis determining factor can be mapped to that position on the sharp arm of the Y chromosome. On occasion, there are individuals who do not have a the Y chromosome, but still are male. That's because this gene has been translocated onto one of the other chromosomes. Now, there are some very interesting implications of this. There are some individuals that doctors must deal with who are not clearly either male or female. On occasion, the doctor has to ask the family, which way would you like this child to develop? There are ambiguities at this time. Some years ago, I had occasion to counsel a young woman with Turner syndrome. This is a deficiency of a Y chromosome and having only one X. This is what she wrote as a poem concerning herself. She uses the term gynandro, and you should know enough Greek to realize that means male, female and male. I'm not a delusion. I'm a gynandro. I'm not an illusion. I'm a gynandro. I'm not a freak. I'm a gynandro. Effected at my conception, not defected by my birth, like you are a woman, or you may be a man. I am a gynandro. And I'm better at being a mixture than I am at maintaining to be one of either extreme, north, south, east, west. Look closely under that microscope of yours with both eyes open wide. You shall see you're wrong, doctor. I'm not abnormal. I followed my instructions perfectly. I'm not a deviant. I did exactly as my chromosomes had set for me. And she goes on in this poem, which I thought was a very poignant expression of the problems of a person with ambiguous sexual development. What does this have to tell us elsewhere about development? Some years ago, there was this book called In His Image, which doesn't mean what you thought. It purports to be of a man who wanted to have a son in his own image. And it claimed to describe human cloning. We now know that the book was a farce. It was not an actual event. In the upper left, I was the geneticist Anderson who called and tried to check with the publication company, give us the evidence for this, but there was no evidence. I talked about this at another location, and I thought this was a rather nice way to deal with this. This is something which is not likely to be used. The problem is that in an adult cell, from any adult cell that I have, the chromosomes are now programmed to read an adult product. It has so far proved impossible to get an adult nucleus to start back at the beginning. We don't know how to push the rewind button on the tape. The mouse to the right had 6 parents. It is a mosaic. 3 different embryos, each having 2 parents, at about the 8-cell stage, were nudged together. It's a chimera. This indicates that these genes in these various embryos, pre-embryos, were able to work together and bring about development. That female to the right had the young at the left. Apparently the ovary also was chimeric. This is even better, or worse, whichever you wish to say. This chimera is called a geep. Sheep and goat are not even the same species. They're not even the same genus. They don't even have the same chromosome number. But there is a sufficient similarity in the genes that development is possible. It would be technically possible to fuse a human embryo with an ape embryo, if one wanted to use that means to test the degree of genetic similarity. I suggest that that should never be done. This also can be used to do some tracing in history. sickle cell gene is on a different background. And where you see the symbol, the number 7.6, these individuals have the same type of chromosome carrying that gene. Where you see 13, these individuals have the same type of chromosome for a different kind of chromosome that acts as a marker. Perhaps you heard a few years ago about the search for mitochondrial Eve. You remember that? Well, the speculation was not well founded. The sample size was too small. There were some problems with that. But the time will come when we can use information like this to trace much more accurately than we have in the past. I want to stop the slides and conclude very shortly. This is one of my favorite cartoons. And actually, we're in job to do both of these. Now, there are some people who give answers and make assumptions concerning about them when we should not make those kinds of assumptions. So let me conclude with asking, what is this telling us about human nature? What do the sciences tell us about human nature? What are the distinctions that we can make? So let me list some things very, very briefly. First, humans have their own nature and not that of another species. Even though we may find similarities, there are distinctives. Secondly, human behavior taken as a whole is unique. In other species, there are indications of tool making, tool using, and so on, but the combination in the human is not found elsewhere. A third point, humans do occupy a significant place in the universe. Fourth, the human body has an inestimable value. You may have heard the sermon illustration claiming that the chemicals in the human body are worth $1 or 2 or 3 with inflation. But that applies only if the body is reduced to ash. If you think of all of the very complex chemicals, some in very small quantity, there is no way that we could replicate. The last point here is that I feel that we should be grateful for human similarities with other species, since they permit our use of medicines and experimental animals to study human problems and to test for toxic materials. There are some things that the sciences will not tell us, and the scriptures do tell us. And these are well known to you. Human existence was planned by God and was the result of a deliberate creative act. We are not the unplanned result of natural self-sufficient causes. We are God's handiwork. We are in a unique relationship with Him. At the beginning of the next hour, I will reflect somewhat on the theme of us being in God's image. And I want to ask you, from what you understand from that, does your concept of humans in God's image really face up to some of these things that I'm talking about? Does it need to? Do we need to retranslate and reapply these? Let me conclude with a few sentences from a meditation from Francis de Sales, this classic about devotional life from some centuries ago. Consider that so many years ago you were not yet in the world, and that your being was a mere nothing. God has drawn you out of this nothing to make you what you are now, merely out of His own pure goodness, having no need of you whatever. Consider the being that God has given you. It is the greatest in this visible world, capable of eternal life, and of being perfectly united to His divine majesty. Comments, these very small part of a window on what's developing in DNA, and encourage you to be thinking about this, to find resource people, to feed this into the way that you consider other questions. Now let me show you one slide, please. You cannot see the artwork to the right. It is imaginative. It is in the most recent copy of a biotechnology journal. But I want to read with you what it says to the left. Recent technological advances made possible by biotechnology have allowed scientists to explore the brain at the molecular level. Neural receptors can now be cloned, and genes associated with Alzheimer's disease and schizophrenia have been identified. Now notice, venture capitalists have been providing the funding for neuroscience startups to develop new technologies that might be used to solve the mysteries of how the brain functions and why it sometimes fails. I'm glad that money is being put into this kind of work. The study of the human brain is a very important frontier. It is possible, as this information accumulates, it could be misinterpreted and misapplied. Now, for a few minutes, I want not to show the slide, but to digress. Bruce Reichenbach is at Augsburg College in Minneapolis in philosophy. He and I put together a paper in which we tried to reflect on the relationship between the sciences and our views of imago Dei. It's not my specialty. It's not something I want to tell you about where you've studied for a long while. But let me try to bring together a very short review of some of the impressions of what it means to be in the image of God. The point here is that in the scriptures, a variety of points of view are used. This should not be surprising because the scriptures do not present a human psychology, biology, sociology, or anthropology. They're talking about something else, about our relationship to God. It is possible, however, to distinguish 4 different interpretations. And as I go over this in a very outline-ish form, you can try to ask yourself, will anything that I say in these 3 lectures affect the way that you think about imago Dei? One is the ontological or substantive interpretation. Augustine, for example, held that the imago Dei refers to the rational soul and specifically to its unique abilities of memory, understanding, and will. Now, there are reasons for this. Since the imago Dei designates a unique feature of human persons, it must relate to the essence of man. That which distinguishes humans from animals is rationality and the ability to will and to act morally. Hence, the imago Dei must be identified with something of that sort. Also, since God is spirit, that which images him must likewise be spiritual. Under this view, the imago Dei is found in the soul and not the body. A second view locates the image not ontologically, but in the human moral status before God. According to Martin Luther, humans were created holy, morally perfect, a perfection which was lost in the fall. According to a third view, the imago Dei is relational. It reflects, in part, the male-female distinction that was made. Relationship is the key to understanding. And finally, some have held that the image is functional. delegates his power to humans, his power by virtue of creation. And thus, our dominion over the earth reflects God's intimate lordship. Now, I would suggest that over the years, these varying points of view have reflected in part the worldview, the other things that were happening at the time that these ideas have been developed. And I do not know. I'm simply asking you to think. As we attempt to keep this concept relevant for the present time, does the information that I'm talking about affect that at all? And thus, I'll be talking about the brain and our information about genes and the brain. The brain has about 2 to 3% of the body weight, and yet up to 50% of oxygen utilization. The development of the brain is pre-programmed in great detail. Cells divide and then migrate. Some cells are pre-programmed to die. Of more interest to me at this point is that the brain is different from other parts of the body. All cells, with few exceptions, in the body have the same genes, but only a small fraction, about 5%, is transcribed. All the DNA, the messenger RNA, is only about 5% of what it could be. That is, in the liver and the rest of the body. But in the brain, about 20% of the DNA is expressed. Let's look at it a different way. 50,000 genes. Let's imagine that we have a pie that's divided up into 6 parts. 2 pieces. Those genes are expressed only in the brain. Another piece expressed in the brain and elsewhere. Half of the genes that we have expressed in the brain. Sometimes with a very small product. The expression may differ at the time of development of the brain in various cells within the brain. All of this is beginning to come to light. There are genes for receptors that recognize neurotransmitters. There are genes for channels that regulate the flow of ions. It is amazingly complex. This is the basic dogma for genetics. The DNA at the far left can replicate itself as cells divide. This is transcribed into the form of the message in the RNA. This is translated into a product. This shows one of the kinds of genes expressed in the brain. In this instance, a product is made, and it is differentially cut up after it has been made. You may recognize ACTH. You may recognize to the lower right endorphin. MSH is melanocyte-stimulating hormone. So that it's a very economical process. The gene makes a product, and it can be cut up into different functional units. It's also possible to have alternative splicing, where given a long gene, sometimes you'll read out a part of it, sometimes a different part, and sometimes a different part yet. This adds to the complexity of the brain. This particular gene is located on chromosome 2. This is identified by applying a radioactive label to the chromosomes and simply observing which chromosome has more green spots that develop from it. This is not a genetic picture, but it illustrates some developments in brain imaging. These are PET scans, positron-emitting tomography. The individual is given an injection of a very low-level isotope. The individual is asked to read the level of activity. Here, this represents the difference in the activity of the brain in terms of words. You notice to hear a word, to see it, to speak it, and to think about it in the sense of asking for what synonyms there might be. Different parts of the brain are used. Facetiously, I've thought, would this help us to know if a religious thought is different from a scientific thought? Is a different part of the brain used? It's not impossible to raise that question. More recently, this has been done with a different kind of isotope, which can be measured. 10 minutes later, you can re-inject and measure again. Some individuals that have severe depression show a hot spot, a high level of oxygen use and of blood flow in one particular part of the brain. You can speak to these individuals and say, we know how your brain responds. We want you now to think happy thoughts. That hot spot disappears. Or you can talk to a normal individual and say, I want you to think the saddest thought you can. The spot appears. So there are ways of modifying. This new opportunity for getting imaging of the brain will change our views. This is a complex diagram. The details are not important, but I want to give you some reflection about it. The term artificial intelligence has been used. This is given the idea that someone may try to use computers to see if you can copy and mimic what brain action is carrying out. It is thought, for example, that if you can design a computer program which can do all the kinds of things that a human can do, in a sense you have shown that the human is not necessary. But people are not talking about artificial intelligence in that sense currently. There is a second use to try to have computer programs that will do something of use, which may or may not be like a brain activity. That is, to be able to hear speech and convert it into typed words. Computer programs are very far advanced in this. But to me, the more interesting aspect is what's called computational neuroscience. That is, to use the brain, to use the computer to try to understand how the brain works. This has to do with studies of imaging, of vision. It is quite clear that there are separate areas of the brain that function for form, for color, and for motion. So there is a segregation of function. But there also will have to be means for integration. This is the kind of diagram that now emerges. Each of these parts has a different function, but then there are diagrams or networks that read across, that read up and down, backwards and forwards, which permit this kind of integration. This has given rise to the idea of parallel processing. And as a matter of fact, some computer programs have been adapted to use parallel processing. So we're getting a much clearer idea as to how the brain works in this particular kind of way. Now a bit about twins. These are extremely interesting. What do they tell us about the brain and behavior? I've indicated that these can be monozygotic or identical pairs, or they can be dizygotic or fraternal pairs. Both can be affected, concordant, or only one can be affected for a particular trait. Perhaps you've heard something about the study of identical twins reared apart at the University of Minnesota. fingers, they fell down the stairs at age 15, they had a miscarriage in their first pregnancy. One pair of twins stored rubber bands around their wrists, cleared the restaurant tables of all extraneous items when they went there, flushed the toilets before using, and fell asleep in front of the TV. Now, you say, that's much too much. I mean, I don't believe it. In a sense, it's also trivial. One side point would be to say that some of the things that bother you about your roommate or your spouse or your teacher may in fact be genetically conditioned idiosyncrasies, and we should be more understanding of each other. I attended a twin congress in the Netherlands, and here is a pair of twins who together run a travel bureau in Omaha, Nebraska. They were delightful people. What about the EEG of twins? Each of these little curves represents a spectrum. You go from very low to very high in terms of the frequency of the background wave. So one that peaks at about 9, most of the brain waves are at that frequency per second. Twin A at the top, twin pair A at the top, are quite different from each other. The third down are fairly much like each other. The bottom one very much unlike. These are fraternal twin pairs. What do you think identicals are? We do not understand fully what it means, but it very likely does have some implications for personality and behavior. We can take a pair of identical twins at 2 different times. Twin A and twin B sample 1 time, sample 2 times. One twin is as much like the other as like itself at a later time. We have a very fascinating pair of twins. The 2 women to the right there have a condition described as reading epilepsy. At about age 17, both of these developed seizures where they would read. There were some warning signs. They would then develop a convulsion, and then they would recover from this. It is not a simple eye movement phenomenon. It is very complex. For example, it is triggered by strange material and not by common material. One of these twins would get a seizure when she read the 24th Psalm, but not the 23rd. Now some other conditions. This is a case of self-mutilation. It is an X-linked gene. It is an enzyme deficiency. It leads to mental retardation and this habit of chewing and biting. This person looked normal at birth. Here are 2 twins that have become interesting to me recently. They have a different kind of face. They have a kind of floppy movement. They're called the happy puppet. They laugh and they smile. They're very pleasant. They're quite retarded, and they frequently have seizures. It appears now to be the result of a missing part of chromosome number 15. These individuals, at the points marked there, have what's called a fragile X. The fragile X syndrome. It is one of the most frequent causes of retardation in boys. These are rather poor pictures of these individuals. It is important for us to understand that and try to know what the results would be. I think you might understand that in the case of one who is a teacher in special education, it might be extremely important to be able to adapt to this and understand the implications. In this instance, it is X-linked. The gene is carried by a carrier woman. She often is mildly retarded, and the boys are often quite seriously affected. Recently, we've learned a lot about We start with a patient that has Alzheimer's disease. Look at the brothers and sisters to see the risk. Each of these lines is a cumulative risk. That is, a person in the lower line would have a risk of about 1% by age 75, but closer to 9% by age 90. I sorted these into two groups, those in which there was a parent who also had Alzheimer's disease, and where the onset in the patient was early, below age 7. The upper curve then, onset before 70, parents affected, the risk to the brothers and sisters approaches 50%. That gave a suggestion that genetics plays some part in this. We now know that in familial cases of Alzheimer's disease, there is a gene on chromosome 21 that is responsible. But not in all such families. Other families are now showing up and do not appear to be mapped there. In that same, in a gene nearby there, not only is there something that makes the product that accumulates in Alzheimer's disease, but there is what's called an enzyme inhibitor. It's becoming extremely complex. I've done a review article. We do not yet know enough to be able to predict in such families what is developed. One of my own fields of study has to do with convulsions. Febrile convulsions are one of the most genetic forms that we have. In the general population, about 2.5% have had it. And I would guess that 3 or 4 of you here may have had febrile convulsions. I did as a child. We have 4 children. Our son also had this. The rates are higher in Japan than in Rochester, Minnesota. The rates are markedly high when a parent has had this. 2 white bars, the third from the left and the far to the left. The white bars, if a parent had this and there were 3 or more episodes in the initial patient, the risk to brother and sister is about 40%. So I've asked myself, what could be a temperature-sensitive gene? It's of interest then that in the fruit fly or Drosophila there are temperature-sensitive genes. These now turn out to be a change in what is called an ion channel. This is an imaginative diagram. Sorry, this is not imaginative. The next thing would be, this one shows 4 generations of febrile convulsions. We think there may be a mappable gene. We are now collecting families to see if we can locate a specific gene to this. This is a picture now of an ion channel. If you imagine the membrane of a muscle or a nerve, there is a pore, but this pore is controllable by voltage. It then can let sodium in or not let sodium in. It turns out that the gene for this sodium ion channel in the fruit fly is very much like the gene in the rat and very much like one that's in the human. That might sound surprising, but if you think about the fact that an ion channel to function must have a certain structure, this structure is relatively much alike across these species. One of these genes has now been mapped to human chromosome number 2. My job is to find out whether a gene in febrile convulsion families also maps to chromosome number 2. I'll be saying a little bit more now about some other traits that are perhaps more troubling and more puzzling and not as rare. Alcoholism is complex. follow this. The other kind of trait, which has earlier onset, where it's much more likely to have fighting and arrests, there is inability to abstain. This appears to be more genetic. What do you do in a particular situation? It's very difficult at this time to say, this condition is genetic and this one is not. But one must realize that the possibility does exist, and that when we know better how these are brought about, what the mechanisms might be, I think we might be better able to combine medical, psychological, and spiritual therapy to work with these people. But there is this option. A writer here in the city of Chicago has made this description. He's described the brain as the yoke that binds genes and culture. It's a very interesting description. Now let me say some other things that are more speculative. I should add that we have already told you that a gene for manic depressive psychosis has been mapped. It now appears that in some families a gene for schizophrenia can be mapped to a specific gene. There was one particular study which found evidence that extreme shyness in children may depend in part on an inherited variation in threshold of arousal. Here is the question, the speculation. If there is increasing evidence that behavior such as alcoholism, or shyness, or depression may result from a combination of genetic and environmental factors, could this also be true for other kinds of behaviors? What would this have to say about our views as to responsibility for behavior? Whenever we have a pattern of behavior and see that genes are involved, then there can be mutations. Notice that when a person is retarded or psychotic, we set different standards of responsibility for them. My hunch is, my hypothesis, is that we may find some other individuals who are antisocial, asocial, amoral, who in fact have some damage to those parts of the brain that are involved in that particular kind of reasoning. I do not mean to infer that we know that now. But if that were to come about, how would this influence the way that we would approach this question about responsibility? Another question then has to do, does this alter our way of thinking about mind and brain? This has been a poser for many, many years. Are these the same or are they different? How do we approach them? Let me suggest three models of the relationship. The first says that the mind is clearly distinct from the brain. While we are here, they are connected in the sense that the brain is the tool by which the mind interacts with the physical world. The second model, the brain is a more complex level of the organism's operations. Mind is the operation of the central nervous system interpreted at a higher and more symbolic level. The third says the mind is merely structure of DNA and also the components of it. However, by asking a biological question, they resolve the problem. So a question from the top down helped to cut through a difference in level. Will it also be possible that we can begin to use biological information to interpret psychological, perhaps using genetic explanations? When we ask a psychological question, that is appropriate. In terms of personhood, what kind of structure of the brain would be required so that we are able to function as persons? Because personhood certainly resides in and uses the brains that we have. Perhaps one final challenge that we might think about comes from the writing of E.O. Wilson. He wrote a book entitled Sociobiology. I was shocked, puzzled, to open that book some years ago and find that the title of the first chapter was The Morality of the Gene. My field is behavior genetics, and I had not thought of putting it that way. What he said was, we will not have a true scientific ethic until we understand the brain that does ethics. We will not know that until we know the genes that specify the brain that does ethics. I think he's grossly gone beyond his evidence, and as a scientist, I could argue that point. But you're going to see more of this. And what I'm trying to alert you to is to be thinking about the relationships here. What can we say? We are God's hand. We have a treasure in earthen vessels. We are different from each other, and part of that is genetic. Having gifts that differ according to the grace given us, let us use them. But now more pertinently, I must ask, is sin in the genes? Some of my colleagues ask, now would you explain the inheritance of original sin? I respond by saying, I cannot do a genetic study unless there is variability. So you bring someone to me without sin, and I will do the study that you're asking. What I talk to you about tomorrow, about genetic engineering, would it be possible to use genetic engineering to eradicate sin? Is sin in the genes? I don't think so. I can imagine an individual with excellent genes who still can be selfish, who can be arrogant. I do grant you that genes affect our behaviors more than we realize. But I don't believe we're talking at the level of sin. Another question. Fruits of the Spirit are, you can fill in the blank, love, joy, peace, the others. Could genetic engineering be used to improve those? I suspect that the tendency to quick anger is first claimed that humans cannot be understood, interpreted, or analyzed fully apart from their being considered physical beings. Both brain research and genetic research point to the extremely important formative roles our biological heritage plays. Hence, it is appropriate and not anti-theistic to try to explain human persons and their behaviors in physiological terms. However, this claim must be tempered by a realization that though explanation might proceed from the bottom up, complete accounts also require top-down understanding. Humans cannot be understood apart from psychical considerations, for they are purposeful beings who seek to understand and find meaning in their world. This means that the natural sciences cannot be the end of all explanation. Understanding of humans must use all of the insights that we can find, the social sciences, and all the other areas, the humanities as well. Finally, a scientific account of the human person is, from the Christian perspective, incomplete, for it fails to take account of our relatedness to God. No matter what else the Imago Dei concept implies, it surely means that to understand humans, we must see them from the point of view of relatedness to God. It is in these terms that human wholeness and human nature must also be explored. So I try to talk with you very briefly about the brain, the knowledge that we now have, and which we surely will